Hello everyone, myself Ransi Patel and I'm here to present a case study on the topic purification and biological analysis of antimicrobial compounds produced by an endophytic Streptomyces species. So basically this is taken up from a research paper. So that the agenda we are going to discuss about is the introduction, which is the general overview of the topic, the microbial source, the metabolites of interest, separation technique and their importance and the outcome of the purification process. So moving on with the introduction. So basically, this paper describes the isolation and characterization of antimicrobial compounds called plicetin from an endophytic Streptomyces species. So basically, that the emergence due to the emergence of fungal phytogens and drug-resistant bacteria, that is a major challenge in agriculture and public health respectively. So basically, there is an urgent need for new sources of antimicrobial compounds to combat these issues. So this study focuses on exploring the antimicrobial activity of Streptomyces species SPF5 against drug-resistant bacteria, especially against methicillin-resistant Staphylococcus aureus, which is MRSA, and vancomycin-resistant Enterococcus uh, VRE, and fungal phytopathogens. So moving on to the microbial source, and it is basically a bacteria and it belongs to the phylum genus Streptomyces, which is a part of the phylum Actinobacteria. And it was originally isolated as an endophyte from the leaves of Citrus jambieri, Raphilemon. And the strains were identified as Streptomyces species SP5. And its characteristics basically it is a filamentous soil bacterium that grows as a network of branching filaments. And it is capable of producing aerial branches bearing chain of spores. So basically it has chain of spores and it is known for producing bioactive secondary metabolites. And the cultivation was mainly done on starch casein nitrate agar plates. And the spores were preserved in 20% glycerol at minus 20 degrees Celsius for long-term storage. For metabolite production, it was grown in starch casein nitrate broth at pH 7 at 28 degrees Celsius under shaking conditions of 180 rpm for 5 days. And it basically produced antimicrobial metabolites effective against drug-resistant bacteria and fungal phytopathogens. So moving on with the metabolite of interest, and the antimicrobial metabolites were extracted from the culture supernatant of Streptomyces species SPF5 using diethyl ether. This extract showed strong antimicrobial activity against both bacteria and fungi. And the main antimicrobial compound was isolated and it was purified from the diethyl ether extract. And it was identified as plicetin, a nucleoside antibiotic. And it basically belongs to the part, and it is a part of the family ampicillin family. And based on the physical properties, it is a white crystalline needle-shaped compound, and it is soluble in chloroform, methanol, and diethyl ether. And it's sparingly soluble in water. The structure of lecithin was confirmed using various spectroscopic techniques, including LCMS and UV visible spectroscopy and FTIR. And lecithin showed broad spectrum antibiotic activities against drug-resistant bacteria like MRSA and PRA. And it also showed the activity against fungal phytopathogens like Funsaria moxysporum and Alternaria brascoli. And basically, this study was the first to demonstrate plicetin's antimicrobial properties against the specific bacteria and fungi. And its mechanism of action was plicetin causes cell deformation and lysis in bacteria, likely by disrupting the cell membrane integrity. So moving on with to the separation technique, and we'll see their importance. So first is thin layer chromatography. So basically in thin layer chromatography, if I were to give a simple analogy to explain it, imagine you have a sponge which represent the TLC plate and you are drawing a straight line at the bottom. Now you put a tiny drop of different color ink at this line. Now this line basically is your samples. Then you stand the sponge in a shallow puddle of water. This is your solvent at the bottom. As the water climbs up the sponge, it carries the ink color with it. Different color will travel at different speeds and based on how much they like the water versus the sponge. Some of the colored particles will move quickly and go far, while the others will move slowly and will stay at the bottom, close to the starting line. So at the end, we can see how far each color the particle has traveled. This can help us to separate and identify the different parts of the mixture. So in this case study, thin layer chromatography, which is TLC method is used, in which the stationary phase is basically the silica gel, F254 pre-coated plates, and the mobile phase is the ethyl acetate is to chloroform 9:1 volume by volume. The detection methods use the iodine vapors and UV light, followed by bioautography, in which TLC strips are mounted on agar plates and are seeded with the test organisms, which are basically methylene resistant Streptococcus aureus and Fusarium oxysporum. And the incubation period was 1 hour at 4 degree for compound diffusion. And then it was 24 to 48 hours at temperatures. And the result obtained was one of the active compound was detected and the RF value of the active compound was 0.9. RF factor is basically retention factor, which is equal to distance traveled by the compound divided by the distance traveled by the solvent. So moving on forward to silica gel column chromatography. And if I were to give you a simple analogy to explain this, so it is basically like filtering a mixture through a tube filled with sand. So now imagine you have a tall tube filled with fine sand. So this represents the silica gel in the column. And at the top of the tube, you pour a, a mixed bag of marbles of different colors and sizes. So these are basically your sample molecules. And next, you slowly pour in water, which is basically a solvent here, through the tube. And as water flows down to the sand, it carries the marbles with it. However, different marbles will move through the sand at different speeds based on their size and how they interact with the sand. Some marbles will travel quickly and come out of the tube first, while others will take longer to make their way to the sand. By collecting what comes out of the bottom of in different containers, we can separate the marbles based on their speed. So this is what basically happens in silica gel column chromatography. So here in this case study, silica gel column chromatography was used for the purification of antimicrobial compound from diethyl extract. And the stationary phase was silica gel and the columns were initially packed using chloroform and it was followed by elution, which is a chemical process that involves removing a material's ion by ion exchange with another material. And it was initially done using 100% chloroform and gradually increasing ethyl acetate concentration. And it was ended with 100% ethyl acetate. So basically, different ratios of chloroform plus is to ethyl were taken, and the volume for each step was 200 ml. And the total fraction collected from this was 88. And the volume for each fraction was about 25 ml. And the active screening was done for all the fraction and were tested for antimicrobial activity against MRSA and Fusarium oxysporum. And the method used was standard curvivoid disk diffusion method. The result what we got was active fractions of 30 to 40. And the elution composition of the active fraction was chloroform is to acetate 60 is to 40 volume by volume. So moving on to high performance liquid chromatography, which is HPLC. And if I were to give an analogy to explain this chromatography, so it is basically you have to imagine you have a jar with marbles of different colors, and you have to pour these marbles down a board that's tiled. And the board has small barriers and paths that can slow down some marbles, but let others pass quickly. So as the marbles roll down, some colors get slowed down by the obstacle, but while the others move faster. So by the time they reach the bottom, the marbles are sorted by colors because different colors were moving on at different speed. So basically in HPLC, instead of marbles, you have liquid mixture of different chemicals. And the tile board is like a column filled with materials that interact differently with each chemical. So as the mixture is pushed down through the column, different chemicals move at different speeds. So depending on how they interact with the material in the column, at the end the chemical comes out separated and the detector identifies and measures them. So this is how basically what goes on with HPLC. So HPLC here is basically used for final purification of the antimicrobial compound and for confirming the compound purity. And the type here used is reverse phase HPLC. So reverse phase HPLC, the difference between a normal HPLC and reverse phase HPLC is basically that in normal HPLC, the stationary phase is polar and while the mobile phase is non-polar. But in reverse phase HPLC, it is basically the opposite. The stationary phase becomes non-polar and the mobile phase becomes polar. And and it was basically tested against active fractions of 30 to 40, which was obtained from silica gel chromatography. And the four prominent peaks were detected, and the most active peak detected was at 12.25 retention time. So retention time is basically the refers to the time taken by the compound to elute out from the chromatographic column. So the active peak obtained was 12.5 retention time, and when rechromatographed, the active time again obtained was 12.5 chromatograph at same conditions of actinonitrile is to water 95.5. A single peak was obtained, which confirmed the purity of the isolated compound sp5. So finally, we'll discuss the outcomes we have received from the purification process. So here are some graphs two graphs in which graph A represents the chromatogram showing different peaks at different times, wherein the x-axis represents the time it takes for the substance to travel to the chromatograph and reach the detector, while the y-axis represents the intensity of the signal detected, and it is basically measured in milliabsorption units. And higher peak means more of the substance is present. And at peak times, and at 0.246, as you can see, there is a very small peak, and at 3.10 minutes, there is a small peak indicating there is a presence of substance. However, as you can see, there is a more significant peak at 12.255 minutes, indicating a large amount of that particular substance. And graph B, basically in graph B, it represents only one prominent peak. As it is similar to graph A, in which the x-axis represents time and the y-axis shows the signal intensity. The single peak that is shown in graph B indicates a single substance that is present in large amount at this specific time. So basically, the comparison between both the graphs is that graph A shows a complex mixture with many different substances, but graph B shows a simpler mixture with mainly only one substance. And the key substances, both the graphs have significant peak around 12.25 minutes, indicating that the substance is present in both the samples. So this table here presents specific chemical shift data for different moieties of a molecule, and it's comparing values from three sources, which is basically an unnamed primary source and two other sources. And overall, this table allows us a detailed comparison of chemical shift data between different studies, highlighting both consistencies and variation in the characterization of these molecules. The variation could be due to anything like differences in experimental conditions, sample preparations, or data processing methods used by these different research groups. So thank you. This is all I have got for you. I hope you have enjoyed this video. Thank you very much.